Hi, hello everyone, welcome to my channel. In this video, I am going to discuss the topic histology of kidney. Okay, so kidney are the parts of excretory system. Okay, and a person will be having a pair of kidney. Okay, or you can say two in number. The kidney will be two in number. Okay. The kidney are the part of excretory system, and these kidney, what is the function of this kidney? This kidney are mainly associated with the production of urine, okay, through which what are all the nitrogenous waste which are formed in the body are removed out of the body. And the kidney are one pair of organs with a convex outer surface and a concave inner surface, which are present on either side of the vertebral column just above the vast. Okay, so the kidney will be one, it is a one pair organ, means a person will be having two kidney. Okay, and they are present on either side of the vertebral column just above the vast. And you can see the kidney structure will be like this, which is bean shaped structure. Okay, surface is convex and the inner surface will be concave. Okay, and as I mentioned here, the shape of the kidney is bean shaped. You can look at here, it is like a bean. Okay, bean sirutala, like that it will be. And color, the color of the kidney, it will be dark red in color. And length, the length of the kidney will be 10 centimeter long, and the width will be 5 centimeter, and thickness will be 4 centimeter. Okay, thickness will be 4 centimeter, and this one will weigh about 150 grams. The kidney will weigh about 150 grams. And position, where the kidney is located, this kidney lies on the posterior abdominal wall, just below the diaphragm. Okay, now this the inner concave surface. Okay, this inner concave surface has a depression. Okay, this inner concave surface has a depression in the center, that is called as hilus, or you can say that is called as hilus or hilum. Okay, what this inner concave surface has a depression, that is called as hilus or hilum. This is the place where renal artery, so you can look at in the diagram here, this is the place where renal artery enters and the renal vein and urethra leaves. Okay, the renal artery enters and the renal vein and the urethra leaves. Okay, this is the place. Which place? That is hilus. Okay, as I mentioned before, you can look at in the diagram, these are the two kidneys which are present on the either side of the vertebral column, which are bean shaped and they are dark red in color. Okay, so above the kidney you can find out the adrenal glands. Okay, now we will move on to the next point. When you take the vertical section of the kidney, okay, when you take the vertical section of the kidney shows the three parts. Okay, which are those? It shows three parts, that is capsule, cortex and medulla. The first part is capsule, second part is cortex, and third part is medulla. Okay, see, if you take any in general, if you take any cell, okay, if you take any cell, okay, if you think this is the cell, the outer part, okay, this part you can look at here, this outer part actually forms the cortex. Okay, and the middle, okay, this part, middle part, usually what we will call it as, this one will be called as medulla. I'm not mentioning that kidney for kidney purpose, no, it is same for all, any, if you take any cells, if you explain, so this part will forms the medulla and this part will forms the cortex, and the outer covering layer, it is called as capsule. Okay, now we will discuss the one by one now. The first one, that is capsule. The capsule, it is nothing but it is a thin fibrous connective tissue. So as we are studying the histology, means where we are study about the tissues. So look at this, the capsule, that is outer covering. This one, the outer covering, okay, is made up of, it is a thin fibrous connective tissue which surrounds the kidney, which makes the outermost covering, that is called as capsule, or it is called as renal capsule, because the kidney we refer it as renal. Okay, so the covering of the covering, outer protective covering of the kidney is called as capsule. Okay, this part is called as capsule. Now the next one, that is cortex. The cortex, it is a reddish brown layer of tissues that is present immediately below the capsule. As I mentioned, this is the cortex capsule. So immediately just below the capsule, the cortex is present. Okay, the cortex is present, and this cortex is consist of again 3 parts, that is Malpighian corpuscles, the second one proximal convoluted tubules, and the third one distal convoluted tubule. The proximal convoluted tubule, it is also called as PCT, and distal convoluted tubule, it is also called as DCT. The next part is the third one, that is medulla. So medulla, you can look at in the diagram here, this region, okay, this region is called as medulla. So this medulla, it is the innermost layer consisting of conical areas called as renal pyramids. You can look at in the diagram here, you are seeing the conical areas, okay. These conical areas are called as renal pyramids, okay. Those called as renal pyramids. These are called as renal pyramids, which are conical in shape, okay. Now these renal pyramids, okay, these renal pyramids are separated by renal column. Is separated by renal column. These renal pyramids are separated by renal column, okay. And these renal pyramids are usually in each kidney. How many will be present? Will be 8 to 18, okay. These renal pyramids in each kidney, there are 8 to 18 renal pyramids in each kidney, okay. And the apex of each pyramid is called as a renal papillae, and okay, if you look at this apex, okay, our projections, those are called as the renal pyramid, our apex, okay, that is those our projections, that is called as renal papillae, okay. And these renal papillae projects into a small depressions, that is called as minor calyx. In plural form, it is called as calyces, okay. And these renal papillae projects into a small depression, those are called as renal minor calyx, okay. And these several minor depressions, that is minor calyces, combined to form a major calyx, okay, combined to form a major calyx. And the major calyx opens into the renal pelvis, and that will leave the kidney as the urethra. You can look at in the diagram, this part forms the minor calyx, and these several minor calyx combine to form the major calyx, and major calyx leads to pelvis, and pelvis leads to a urethra, which leaves out of the kidney, okay. So in a simple flow chart, you can remember like this: minor calyx, major calyces, and then renal pelvis, and then urethra. So, and the next one, the important one, is nephron. Okay, here nephron, it has another name. These are also called as uriniferous tubules, and this each kidney is made up of numerous secretory units, that those are called as nephron. Okay, and these nephrons are structural and functional unit of kidney. Why these are structural and functional unit of kidney? Because this is the place, this is the structure where the blood is filtered and the urine is formed. Okay, this is the place where, this is the structure where the blood is filtered and urine is formed. Okay, that's why these nephrons are called as structural and functional unit of kidney. Okay, and if you look at the types of kidney, there are two main types of there are two main types of nephrons. Okay, in that the first one is cortical nephron, second one is juxtamedullary. Okay, and this each nephron is divided into again two parts, that is Malpighian corpuscle and then tubular part. Okay, as I mentioned here before, okay, in the cortex region, here I just mentioned what you can find out in the cortex region. Okay, so in cortex region, you are going to find the Malpighian corpuscles, proximal convoluted tubule and distal convoluted tubule. These are nothing but the parts of the nephron. Okay, so this is where you are going to find the nephron. Okay, so this each nephron is divided into two parts, that is Malpighian corpuscle and tubular part. Tubular part is nothing but the PCT and DCT. Okay, now let us look at the first one, that is Malpighian corpuscle. The Malpighian corpuscle is composed of double-walled cup called as Bowman's capsule. You can look at in the diagram here. So this Malpighian corpuscle, it is composed of double-walled. It is composed of double-walled cup shape. If you look at this structure, you can see it is like a cup. Okay, it is like a cup-shaped structure. Okay, that is called as Bowman's capsule. Okay, and this Bowman capsule containing a network of capillaries that is called as glomerulosa, which is lined by endothelium. You can look at in the diagram here. It is made up of blood capillaries. Okay, network of blood capillaries. Okay, that is called as glomerulosa. And here the blood capillaries from where the blood is entering. Okay, that part is called as afferent arteriole. Okay, what it is, from where the blood is entering. Okay, that is called as afferent arteriole. Okay, which will be broad when compared to the efferent arteriole, because this is the where the blood is going to live, and it is very narrow when compared to the afferent arteriole. Okay, and this one is lined by squamous epithelium. Now we will move on to the next points. Okay, the tubular part is divided into again few parts, that is, as I mentioned before, that is proximal convoluted tubule, descending loop, Henle's loop, ascending limb, and distal convoluted tubule. So you can look at in the diagram, this is the complete picture of the nephron. Okay, and I have explained this part. This is the Bowman's capsule. Okay, and it is a cup shaped one, and it has a blood network of blood capillaries that is called as glomerulosa, and from where the blood enters, that part is called as afferent arteriole, and where the blood is going to live, that is called as the efferent arteriole. Okay, and this part, it is called as proximal convoluted tube, and if you look at here, this is called as descending limb, and this total huge shaped structure, okay, this huge shaped structure is called as Henle's loop, and this part is called as descending limb. You can observe in the diagram, or you should make a keen observation, this is very thin, okay, and this is little bit broad here. So this part is called as the distal convoluted tubule. Okay, and here there is a ಏನು ಸೆಕ್ರೇಟ್ ಆಗಿರುತ್ತೋ ಅದನ್ನು ಕಲೆಕ್ಟ್ ಮಾಡೋಕ್ಕೆ ಡಕ್ಟ್ ಇದೆ. ಸೊ ಆ ಡಕ್ಟಿಗೆ that duct is called as collecting duct. The secrete, all the secreted or which you have to waste materials have to remove, no? So that have to be collected, no? So that duct is called as a collecting duct. So now the Bowman's capsule having this Bowman capsule, okay, having an inner visceral, this Bowman's capsule having a inner visceral layer. So here what it will be having, it will be having inner visceral layer and an outer parietal layer. It has an outer parietal layer. These both the layers are made up of squamous epithelium. You can look at in the diagram, this is the visceral layer and this is the parietal layer. Both the layers is made up of squamous epithelium. Now, so there are some more terms to explain. That is podocytes. See, if you whenever heard the word CYTS, cytes means cells. Okay, so podocytes are specialized epithelial cells that cover the outer surface of the glomerular capillaries and functions as filtration barriers. So you can look at in the diagram here. So this is the glomerulus blood capillaries, right? So these are the glomerulus blood, these are the blood capillaries. Okay, so these blood capillaries, the outer surface of these blood capillaries have some cells. Okay, have some cells, will have some cells. Those are called as podocytes. Those cells are called as podocytes. Okay, and what is the function of this podocyte? These podocytes act as a filtration barrier. Barrier means obstruction, which is going to ತಡೆ ಅಂತ ಹೇಳ್ತೀಲ್ವಾ, so mix ಆಗಬಾರ್ದು. So these will act as a filtration barrier. Now we will move on to the next part, that is proximal convoluted tubule. Here this proximal convoluted tubule is lined by low cuboidal or pyramidal cells with the indistinct cell borders, okay, which are made up of indistinct cell borders. So if you look at here, if I draw, this is the Bowman's capsule, proximal convoluted tubule. So this proximal convoluted tubule, this is lined by cuboidal cells. Okay, this is lined by cuboidal cells which have the microvilli. Okay, and the nucleus will be at the base. The nucleus will be located at the base. Okay, this is lined by cuboidal cells and the nucleus will be present at the base. So nucleus lies towards the base and free ends of the cells bears a microvilli. As I shown here, this bears the microvilli. These are called as microvilli. Okay, what is the function of this microvilli? Microvilli increases the surface area for absorption. Okay, this will increases the surface area for absorption. Okay, and which how it will looks like? It looks like a brush border. Okay. So, and because of the presence of the microvilli, this lumen of this proximal convoluted tubule looks very narrow. Okay. Now, the next point, that is distal convoluted tubule. So, the next part is distal convoluted tubule. Here, if you draw like this, then this distal convoluted tubule, again this one is also made up of cuboidal cells. This one is also made up of cuboidal cells, but the microvilli will be absent. Okay, if it is present only very less, 1 or 2 you can find out, but the microvilli is absent. That's why it doesn't show any brush border appearance, and also there is absence of microvilli. That's why the lumen will looks very large. Okay, that lumen will looks very large. This lumen will looks very large. Okay, the next one, the medulla. In the medulla region at this point, okay, so from here, if you imagine here, you can see the from like this, it is the Bowman's capsule. Okay, like this it will spread out like this. Okay, like this it will be present. So that's why I mentioned this in this part, cortex region. You can find the what one? You can find the Bowman's capsule, proximal convoluted tubule and distal convoluted tubule. In medulla, in the medulla region, okay, in this part, in the medulla region shows the straight sections. Medulla shows the sections of the straight parts of the tubules. Okay, so that is this descending limb. So this is the descending limb. Okay, this descending limb is lined by low cuboidal or pyramidal cells. Okay, with brush border. Okay, these will be with brush border, means these will be having the microvilli. Okay, and this whole part is called as Henle's loop, and this Henle's loop is lined by squamous epithelium. And there is one more part, that is this one is the ascending limb, and this ascending limb is lined by cuboidal cells but without brush border. Okay, the last part, that is this collecting duct. You can look at here, this is the collecting duct, and this collecting duct will be made up of which type of cells? This one will be made up of columnar cells. Okay, this one will be made up of columnar epithelium. Okay, this will be made up of columnar epithelium. Which one? That is collecting duct. Okay, now there are few more points which are important, which may ask in the examination. Or the first point here, juxtaglomerular cells or glomerular cells. The afferent arteriole have some specialized cells. Those cells are called as juxtaglomerular cells or glomerular cells. Okay, and these cells are endocrine in function. Okay, these are endocrine in function, and the secretes renin. Okay, what is the function of this renin? This renin acts as it will controls the blood pressure. That is, when blood pressure is low, means the actual function is that renin is going to increases the blood pressure when the blood pressure is low. Okay, which cells produces the renin? That is glomerular cells. Okay, this afferent arteriole, if you can look at in the diagram, this afferent arterioles. Okay, this afferent arteriole have the cells, special cells are called as glomerular cells or juxtaglomerular cells. And next point, at the junction of the thick ascending limb of Henle's loop and the distal convoluted tubule will have some specialized cells. Those cells are called as macula densa. Okay, and these, when you look at in the slide, that these will stains very dark. So at the junction of this ascending limb, and this distal convoluted tubule, so you are going to see specialized cells, okay, those are called as macula densa. Okay. And the next point, mesangium. So what is this mesangium? The mesangium is the thin membrane that gives supporting framework to the blood capillaries, which is located in the junction of the afferent and efferent arteriole and the macula densa of the distal convoluted tubule. So all the blood capillaries which are present at the afferent arteriole and the efferent arteriole, and also the macula densa of the distal convoluted tubule, okay, for all these capillaries, that will give the supporting framework, okay, that is called as mesangium, okay. And this mesangium is made up of mesangial cells, okay, mesangial cells, and these mesangial cells are phagocytic in nature, okay. That's about the histology of kidney. And if you look at in the diagram here, this is the histology of the kidney. This is how it will looks when you look at in the microscope, okay. It will look like this. And how you can identify the parts of the kidney, histological parts of the kidney, when you look at in the slide. So here are some hints that I am going to say, explain. So when you look at in the slide, see, like this, something which is present like this is called as Bowman's capsule, okay. So ಈ ತರ ನೀವು ಸ್ಲೈಡ್ ಅಲ್ಲಿ ನೋಡಿದಾಗ, ಸೊ ಈ ತರ ಕಾಣಿಸುತ್ತಲ್ವಾ ಸ್ಲೈಡ್ ಅಲ್ಲಿ ನೋಡಿದಾಗ, ಸೊ ಆಲ್ ದಿಸ್ ಹಿಯರ್ ಈ ತರ ಫಾರ್ಮೇಶನ್ ಆಗಿರುತ್ತೆ, ಈ ತರ ಗ್ಯಾಪ್ ಇರುತ್ತೆ, ಈ ತರ ಲೈನ್ ಅಲ್ಲಿ ಇದು ಗ್ಯಾಪ್, ಇಲ್ಲಿ ಗ್ಯಾಪ್ ಇರುತ್ತೆ, ಅಂಡ್ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಸೆಲ್ ಲೈಕ್ ದಿಸ್, ಡಾರ್ಕ್ ಪಾರ್ಟ್ ಲೈಕ್ ದಿಸ್, ಓಕೆ. ಸೊ ಇದನ್ನ ನಾವೇನಂತ ಕರಿತೀವಿ, ದಿಸ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ Bowman's capsule, okay. So like this you can see, this is forms the Bowman's capsule like this. Numerous you are going to see the Bowman's capsule, okay, Bowman's capsule. So sorry, by mistake I had drawn large one here. So this no slide ಅಲ್ಲಿ ನೋಡಿದಾಗ ಈ ತರ Bowman's capsule, so and you can easily find out numerous Bowman's capsule. That's how you can identify from ಇವಾಗ ಟೆಸ್ಟ್ ಎಕ್ಸಾಮಿನೇಷನ್ ಅಲ್ಲಿ, ಪ್ರಾಕ್ಟಿಕಲ್ ಎಕ್ಸಾಮಿನೇಷನ್ ಅಲ್ಲಿ, ಸೊ ಅವಾಗ ಎಷ್ಟೊಂದು ಸ್ಲೈಡ್ಸ್ ಗಳನ್ನ ಡಿಸ್ಪ್ಲೇ ಮಾಡಿರ್ತಾರೆ. ದೇರ್ ವಿ ಹ್ಯಾವ್ ಟು ಫೈಂಡ್ ಔಟ್ ದ ಎಕ್ಸಾಕ್ಟ್ ಸ್ಲೈಡ್. ಸೊ ಯಾವ್ದು ಕೇಳಿದ್ರೆ ಅದನ್ನ ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು. ಸೊ ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ, ಇಫ್ ಯು ಫೈಂಡ್ ಔಟ್ ಸಮ್ ಸೆಲ್ಸ್ ಲೈಕ್ ದಿಸ್, ಆಬ್ವಿಯಸ್ಲಿ ದಿಸ್ ಇಸ್ ದ ಕಿಡ್ನಿ ಸ್ಲೈಡ್, ಓಕೆ. ಸೊ Bowman's capsule ಈ ತರ ಕಾಣುತ್ತೆ. ಸೊ ಇಲ್ಲಿ ನೋಡಬಹುದು ನೀವು. ಅಂಡ್ ದಿಸ್ ಪಾರ್ಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ mesangium. These are called as glomerulus, okay. And this one is called as podocytes, okay. And you can see like something proximal convoluted tubule, okay. Proximal convoluted tubule ಈ ತರ ಕಾಣುತ್ತೆ. ಸೊ ಈ ತರ ಇರುತ್ತೆ ಅದು ಪ್ರಾಕ್ಸಿಮಲ್ ಕನ್ವಲ್ಯೂಟೆಡ್ ಟ್ಯೂಬಲ್. ಸೊ ಸೆಲ್ಲು ಬೇಸ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ಸ್ವಲ್ಪ ಡಾರ್ಕ್ ಆಗಿ ಸ್ಟೇನ್ ಆಗಿರುತ್ತೆ ಈ ಸೆಲ್ಲು. ಯು ಕ್ಯಾನ್ ಸಿ, ಯು ಕ್ಯಾನ್ ಈಸಿಲಿ ಐಡೆಂಟಿಫೈ, ಬಟ್ ಇಟ್ ಈಸ್ ಮೋರ್ ಕ್ಲಿಯರ್ಲಿ ವಿಸಿಬಲ್ ಇನ್ ಹೈ ಮ್ಯಾಗ್ನಿಫಿಕೇಶನ್, ಓಕೆ. So these cells you can find out, and also there will be long cells like this. Long cells kuda present irutte. So long cells ali, these long cells will be some, it may be Henry's loop or it may be collecting duct, but in low magnification you can't identify. So ee ತರ long cells irodu ನಾವು collecting duct ಅಥವಾ Henry's loop ಅಂತ find out ಮಾಡಬಹುದು. Okay, this is the histology of kidney. I hope you guys learn something from this video, and if you like this video, please do subscribe and share. Thank you everyone. Happy learning.